So today we're going to be talking about muscle contraction. In muscle contraction, you can see here there's a neuron and a plasma membrane which are bordering each other in a synaptic cleft. So usually across a plasma membrane, there's a membrane potential of around -40 to -70 millivolts inside the cell because 3 sodium, which are positively charged, are pumped out, while 2 potassium, which are also positively charged, are pumped in. So whenever your brain wants to trigger a response of a muscle contraction in one of your muscle cells, these neurons have vesicles travel down them, which are filled with acetylcholine. Acetylcholine travels down the neuron and then is exocytosed once it reaches the end, and it travels a short distance until it runs into a ligand-gated channel, which once it has been bound by this acetylcholine, it opens up, allowing sodium in and potassium out, which throws off the normal membrane potential. The influx of positively charged sodium travels along the plasma membrane, running into voltage-gated sodium channels, which opens them through repulsion of the positive charge opening the gate, which in turn creates an even stronger influx of sodium into the cell, because this channel is selective for just sodium and no potassium is leaving. So it creates an even stronger positive membrane potential inside the cell, which flows down the plasma membrane, repeating this cycle by hitting more voltage-gated channels, and it flows down the plasma membrane until it runs into a T-tubule, which carries the charge down to the sarcoplasmic reticulum. The sarcoplasmic reticulum is in charge of calcium sequestration, which creates a huge gradient usually. But this positive charge from the sodium, which has traveled down the T-tubule, interacts with calcium channels, opening them, and it's almost as though a dam has been broken, allowing a huge flow of calcium out of the sarcoplasmic reticulum and into sarcomeres, which make up muscle fibers. Sarcomeres are made up of actin helices with myosin in between, with heads on either side. The outsides are positively charged, the outsides of the actin, that is, and the inside of the actin helices, or the inside towards the middle, is negatively charged. The heads are pointed towards the positive charges at first. The most important thing to remember is that myosin does not condense. Actin helices are made of smaller actin monomers, which each have myosin binding sites on them. However, these binding sites are blocked by tropomyosin. Calcium, however, comes in and binds with troponin, which in turn interacts with tropomyosin, causing a shift which exposes the myosin binding sites. The tropomyosin is seen here in black, and as you can see, it shifts over, exposing the binding sites, and allowing myosin, which is bound with ADP and phosphate, to bind to the myosin actin binding sites. This binding causes a conformational change, leading to phosphorylation or dephosphorylation, or the leaving of the phosphate group, which in turn also causes a power stroke, or the moving of the myosin head more towards the myosin body. This conformational change causes ADP to leave, which causes dissociation of the myosin from the actin-myosin binding sites. ATP then is able to bind to the myosin head, which is again hydrolyzed into ADP and phosphate and put into those 2 binding pockets that it started in, which causes a return to the relaxed position, or basically an undoing of the power stroke. Then it can rebind and repeat the cycle, but this time on the next actin monomer. This causes the whole actin chain to be moved slightly more towards the middle, but because the myosin stays the same length, pulling the actin closer together, the actin keeps moving closer and closer together, pulling on the outsides of the cell and pulling the cell closer together or contracting it.